Hello, everybody. My name is Daniel Ramon. I'm the Vice President for R&D in Health and Wellness in Archer Daniel Midlands. And during the next few minutes, I would like to share with you the results that we have with our probiotic strain, BPL1. All of you know that obesity is a growing concern. It's defined by the World Health Organization as an escalating global epidemic. And of course, obesity is a multifactorial disease in which the diet and the style of life is very, very important. But each time we have more and more data that indicates that genetics and the gut microbiome also play an important role in the development of obesity. Speaking concretely about gut microbiome from the results of Jeff Gordon in the University of Washington, we know for several years ago that there are changes in the gut microbiome of obese people and that there are increases and decreases in some specific groups of bacteria. Taking into account these results 10 years ago, we decided to start the screening of our probiotic collection, searching for a strain that will be able to reduce fat deposition. To do that, we use our animal model, that is the Wolrd Xenobarditis elegans. It's very easy to measure fat deposition in elegans. We only need to stain with red Nile. The red Nile is deposited in the fat drops. As much fat you have, more red Nile is deposited. And by spectrophlorimetry, we can quantify the level of fat. Following this approach, we select from our collection of 1,500 strains, a strain that we call BPL1, that is a member of the Bifidobacterium animalis subspecies Lactis species, that is able to reduce, as you can see at the right of this slide, 42% the deposition of fat. After that, we evaluate the food safety of these microorganisms following the World Health Organization guidelines, including in vivo and in vitro assays. And also, finally, we fully sequence the genome and no concerns related with the food safety of this strain was detected. Also, we optimize the scale-up and industrial production of this strain. And we follow our studies in C. elegans, feeding the worm in the absence or in the presence of BPL1, with exciting results that you can see in this slide. When we run transcriptomic and metabolomic assays, we detect that the animals fed with the probiotic strain have an important increase in some pathways that are related with obesity, like, for example, energy metabolism, feeding behavior, oxidative stress, inflammation, or lipid metabolism. We confirm all these results using mutants in elegans in any one of these pathways. And it's very, very interesting, the strong antioxidant effect of this strain. All these results, that was the first package of information, were published in Journal of Agricultural and Food Chemistry. In the middle of these experiments, we discovered that if we treat the animal, if we treat the probiotic strain BPL1 121 Celsius degrees for 20 minutes, in other words, we kill the probiotic strain, we have exactly the same effect. This was very exciting from a scientific point of view, but also from a technological point of view, because of course we have a probiotic strain that is working in both living and heat-treated forms. And then we move to our second animal model that you can see in this slide, that was we start rats with cafeteria diet. In this model, we can mimic a metabolic syndrome. We use, in this case, both the living and the heat-treated cells of BPL1. And as you can see in the slide, after several weeks of ingesta of BPL1, we detect the same changes in the previous reported pathways in elegans, satiety, oxidative stress, and also fat metabolism. But in this case, also we detect an improvement in mesenteric adiposity and dyslipidemia, an increase in energy expenditure and lean mass, and an improvement in insulin sensitivity. And then we move to the third animal model that you can see in this slide, that is sacker rats. As you probably know, this is a model of hyperobesity. And again, like in the previous cases, we detect a positive effect in the fat metabolism, in satiety, and also in oxidative stress. But also in this case, we detect, I insist that it's a case of extreme obesity, a reduction in body weight that was statistically significant, and also a reduction in the ratio between total cholesterol and HDL cholesterol, as you can see at the right of the slide. Both the previous results and these results were published in 2 journals. And finally, with all these results, we moved to a clinical trial with human volunteers with a slight increase in waist circumference. And in this case, we used 3 arms. One group received placebo, the other one received the living cells of BPL1, and the last one received the heat-treated cells. And as you can see in the slide, after the ingesta 3 months of both the living and the heat-treated cells, there is a statistically significant decrease in waist circumference, and using magnetic resonance, we detect that this reduction was due to the loss of the position of fat at the visceral level. These results were published in International Journal of Obesity. Actually, we have a second clinical trial that you can see in this slide. This trial was done with children with a rare disease that is called Prader-Willi syndrome. This is a disease in which the children has problems of obesity, diabetes, and also mental behavior. And you can see the design of the clinical trial. It's a randomized crossover design in which the children receive the first group during 3 months placebo, and the second group the BPL1, the living cells. Then after this period of 3 months, a period of 3 months of washout, and finally the last period of 3 months in which the first group instead received placebo, received BPL1, and vice versa in the second one. And well, in general, we analyzed as main outcome the change in adiposity using X-ray dual density absorptiometry, and as secondary outcomes, lipid and glucose homeostasis, hyperphagia, and some mental health symptoms that these children have. And here you can see the results. First of all, we detect a significant decrease in abdominal fat mass after the consumption of BPL1 during the period in which the children are receiving the probiotic strengths in comparison with the period in which they are receiving placebo. Second one, we detect a significant decrease in OMA index and insulin, as you can see in the slide, again, during the period in which they are receiving the BPL1 cells. And finally, I can say you that also we detect an improvement in social behavior. All the results has been very recently published in the journal Nutrients. I would like to return with the idea that this strain is working in both the living and the heat-treated cells. And of course, when we are working with the heat-treated cells, we are speaking about postbiotics. This is no doubt about that a new generation of biotics that is arriving. You can see in this slide several papers during the last weeks that appears in different journals that are related with the concept of postbiotics. Postbiotics are defined as soluble factors, products or metabolic byproducts that are secreted by the living bacteria or released after the bacterial lysis, and includes enzymes, peptides, teichoic acids, peptidoglycan-derived monopeptides, polysaccharides, cell surface proteins, and organic acids. Of course, independently of the kind of metabolite, all these postbiotics confer a health benefit to the host when consumed in adequate amounts, and you need to demonstrate this positive effect through clinical trials. Generally are considered as sterile ingredients for a manufacturing standpoint. That means that there are less than 1,000 colony-forming units per gram of the final product in the heat-treated or in the inactivated product. And for postbiotics, I insist that consisting in inactivated microorganisms, the quantification is really very, very important. Most of the cases, the quantification can be performed using flow cytometry. This is the case in our strain. The main advantage of postbiotics is that they are offering flexibility to be incorporated into food and beverage matrix that suffers thermal treatments. And as you well know, this is very frequent in the food industry. There are products easy to standardize. And finally, the longer shelf life and easier to store is another advantage of the use of postbiotics. It's clear that in comparison with traditional probiotic bacteria that are living microorganisms that in adequate amounts confer a health benefit, in this case, postbiotics doesn't need to have a life, but of course they need to procure a health benefit. And in our company, in ADM Biopolis, we would like to lead the way in postbiotic development with this strain, the heat-treated BPL1, that possess a known and patented mechanism of action. You can see at the right of this slide the results that was published by Mintel 1 year ago about the consumer interest in biotic terms that is clearly growing, with 141% in probiotics and 83% in prebiotics and 91% in postbiotics. And this is mainly because you can use these postbiotics in non-traditional applications, like, for example, food beverages or dietary supplement powders. But I insist, you need to maintain the activity, the functionality in the non-viable cells. And this is very clear for our probiotic BPL1. I return to the results in our clinical trials using the living and the heat-treated cells. And as you can see in the bottom of this slide, the heat-treated BPL1 had significant reduction of waist circumference during the 3 months, around 2 centimeters. It's also a reduction of waist circumference versus the height ratio that is statistically significant. The same is true for conicity index, for the visceral abdominal fat, the diastolic blood pressure, and the reduction on the OMA index. All this with the postbiotic, with the heat-treated BPL1. In other words, BPL1 is the world's first postbiotic targeting visceral fat reduction and metabolic health, is easily incorporated into dietary supplements and food and beverage products, is manufacturing friendly, is self-affirmed GRAS, and most important, during the last months, we have identified the molecule of this microorganism that is responsible for the functionality, for the reduction in fat deposition, and is a component of the cell wall. Of course, it's possible to use the heat-treated BPL1 in a lot of applications in food and beverages and in dietary supplements. You can see some of them in this slide, and I insist, it's self-affirmed GRAS, it's QPS listed in the European Union, it's non-GMO, it's organic compliant, it's free of allergen labeling, it's clean label, it's gluten-free, it's kosher, and it's vegan. And finally, I would like to remember you that Bifidobacterium lactis BPL1 was awarded with the Nutri-Ingredients Awards 2020 for the best ingredient of the year in waste management. And the jury said that this is because the strong scientific dossier in the strain. Actually, we have 9 publications in Science Citation Index that covers all the results that I showed you in my previous slides. Thank you very much.